Hi, I'm Audie, and I'm a sophomore at St. Mary's.
Hi, I'm Greta, and I'm a freshman at St. Mary's.
And it's our pleasure to introduce Misha's father and Lucy's son.
In 2007, Nobel laureate Mario Capecchi.
Hello, I can be heard in the back?
Yes.
Way back? Yes, perfect. It's my pleasure to be here and sharing our work with you. My talk will have three parts. The first part, I'll give you a brief description of what we do and why we do it. And then the second part will be more embarrassing. It's sort of an autobiographical sketch. And then the third part, hopefully, will be fun. It's about a trip to Stockholm. Do I have a pointer?
No, just gotta click. You just gotta click on your computer.
Oh, we don't need it. What? Okay, that's fine. That's good. Good. So, the first question that you might ask is, what do I do as a scientist? And now, you just had the students make this structure in all sorts of creative ways. And it's the basis of everything that we do. It's a beautiful structure, and it's an amazing structure because right away as you see it, you can see how it works as an archive for information. And this is information about you. Every one of our cells has an extremely complicated instruction manual. The instruction manual is 3 billion base pairs. It's letters A, C, G, and G. Rather than having 26 letters, the text is written in 4 letters. And it's the order of those letters that provide the information. And this structure shows that 2 strands, one's colored green and the other one's colored red, so you can distinguish those two, and they form a helix. And what's in the outside of the helix is what's holding everything together, the backbone of the DNA. And then the bases, the information base, are inside, and they're forming the steps. But what's amazing about this molecule is, first of all, who originated it, and this is Watson and Crick. Just after they came up with the structure of DNA. Watson was 25 years old. Crick was around 40. And you're showing them with the structure, but the person that's also should have been credited is Rosalind Franklin, who actually provided the structure, the crystallographic image of DNA upon which the model is based. Okay. And Rosalind's mentor showed Watson this structure, this figure, and Watson had a photographic memory. He looked at it and that captured it, all the details, and then was able to describe it to Crick, and Crick right away said, wow, it's a helix, just from looking at that structure. And that set him off in the right direction. I should point out, the other part of it that was important about the structure is that it's what's shown here, is that if you have C, the letter C on one side, the opposite strands will always be G. If one strand is an A, the other letter, then on the opposite strand it will always be T. So A and T are always a base pair, and G and C are always a base pair. And that structure is made such then it's the only possibility of putting the things together so that then they would form an equal space between them, so that then all the strands all the molecules that are holding it together, the backbone, would be equidistant, making the helix. And so it spirals up, but what's beautiful about that structure is simply, whoops, maybe I don't have that next. Okay. We're putting it together as we go along. Okay. So what's important is that the structures those two strands can separate, and then each forms a template for the other strand, because it's always, if there's a T here, the other strand will always be an A. Okay, so here you have two strands can separate. Each forms now a template for making the other strands, and now you've duplicated that information base. And the fidelity of that process is enormous. Imagine being a scribe and have to write out 3 billion letters. This would occupy 1,000 volumes, each 1,000 pages long. Enormous amount of information, and it does so making 1 or 2 mistakes every time it's replicated. Huge fidelity, and that allows your children to be like you. But not quite like you. And that's good too. If they were exactly like you, that would really be terrifying. So, how did we come into the game? We knew about DNA, and pretty soon people were starting to be able to sequence the DNA, and I actually happened to be in the lab where the sequence of about 10 base pairs was first worked out. And it took years. Today, that can be done in a day. All 3 billion base pairs. And the cost is going down enormously. Orders and orders and orders of magnitude. So what our task was, here we have a collection of A, C, and T's and G's, all in a long strand, but we have no idea what it meant. It's like you don't have a Rosetta Stone. You have the language, you have the letters, but you have no idea what the meaning is. And so we took upon ourselves to say, how could we approach that in something as complicated as a mouse? Okay, how could we figure out what all these letters meant? And the idea was very simple. You disrupt this sequence somewhere, go to volume 63, page 379, change that sequence, and then ask what the consequences are. If little finger disappeared in a mouse, then we know we're in a program for making little finger. And methodically now you can go gene after gene after gene, and then start figuring out what the role of each gene is in the formation of a living mouse. So that's what gene targeting is and why we wanted to do it. Okay. And I've just said that. Good. So why do we choose a mouse? And that's shown here. Good. So the reason is that it is a mammal. We are mammals and a mouse is a mammal. So being a mammal, there's many things in common. First of all mammals, their DNA is more than 99% the same in terms of gene content. Okay? So whatever we found out was likely to be true. If we knock out this gene and little fingers disappear, that should happen also in humans. Okay? So whatever we're learning now, finding out in the mouse, is going to be directly applicable to humans. We have a common developmental plan. We have a common, plumbing. It's all the things in your body that have to be put together is the same in a mouse. In your hand, you have over 30 bones. Okay, the exact same set of bones are present in a mouse hand. The only difference in a mouse is we have big fingers and little toes. They switched around, and they have short fingers and big and long toes. Okay? But the plan is identical, bone by bone. And so that's what this says, simply whatever we're going to learn in a mouse is likely to be applicable to humans. And it goes even further than that. You say, well, I do something in the mouse, and it's not doing the same thing in the human. Then it says, wow, this is really interesting, because there has to be an explanation. It cannot just be random. Okay? So whatever we're learning in a mouse is going to be enlightening in terms of the human, whether it's the same or different. And in fact, I can give you many examples where it's different, and that knowing the cause of that difference tells us a lot about humans relative to mice. Okay. So, how do we do gene targeting? How do we change a specific gene in a specific way? We take advantage of a phenomenon called homologous recombination. When we started, we knew about homologous recombination, but it had all to do with sex. That's interesting, but we weren't interested in sex. We are interested in sex, but not for this particular problem. Okay? So, what we found was that the cells that we were working with, these are fibroblasts, skin cells, connective tissue cells, they had homologous recombination. And what is homologous recombination? That's shown schematically here. We have a sequence, the yellow band, that's a DNA strand, and then a blue one. And I've colored them differently simply, but they actually represent the exact same sequence. And what happens is those two strands come together, line up with each other, and then there's literally a break and a re-exchange. So now we've exchanged information. Okay? So you can see there's a break and rejoin. So that now yellow strand is next to the blue strand, and so on, and vice versa. But the sequence has been disrupted. In the process of that exchange, the letters across that sequence is the same. So very precise, and it only happens between like sequences. And that's homologous recombination, and that's what we took advantage of to do gene targeting, which is shown in the next slide. Whoops. Yeah, it is. Okay, good. What I'm seeing here and what I'm seeing there is not quite the same. That's okay. So, the above bar represents your DNA of interest, your favorite gene. The asterisk represents a change in that gene, and we can do that in the test tube. Okay? So then we introduce it back into the cell, and the cell has a machinery that captures that piece of DNA, then searches the entire genome, all 3 billion base pairs, and then finds the same sequence and then exchanges it. But in the process, it has to exchange through like sequences. It can't exchange where the asterisk is, that's different. But in the process of exchanging on either side of that asterisk, you introduce that change that you create in the test tube into the living chromosome of the cell. Okay? So that allows us to change anything we want. And what's remarkable is that asterisk could be anything. It could be 1 letter, it could be 10 letters, it could be 1,000 letters, and we're working on methods to make it millions and billions of letters. Okay? So that we can change the DNA specifically, anything we want, in any gene we want, and then as a process, if little finger disappears, then we know we've changed the gene that's involved in making little finger. And thereby, by seeing what's happening to the actual animal, we can infer what that sequence is doing. Okay? So we infer by what's not happening to the organism. For example, it can change the behavior of the organism. That we're interested, we're doing genes that are important for behavior. Or it can change the structure of the mouse, and thereby we know we're in that particular structure, whether it's a bone or a muscle or the eyes and so on. So it allows us to interrogate any function in a living mouse and therefore infer what that function is also in humans. So in 1980, I submitted a grant saying, wow, this is really exciting, let's do it. And the effect was that the study section looked at it and said, impossible. And the argument was quite reasonable. That little piece of DNA that you've introduced into the cell will never, ever find the same sequence in the genome. 3 billion base pairs, 1,000 volumes each, 1,000 pages long. Would never happen. And there's no way to dispute that. They may be right, they may be wrong. So what did I do? I didn't go crying home and said, they give me money. What I did was simply say, I can still do it. I'll take money from another grant and put it all into this project and see if it goes. It's a little risky. What would happen if you failed? You would have never heard about me. You only hear about the successes. Okay? High risk has risk associated with it, but unfortunately you only hear about the successes. And not only the successes, but the timing had to be perfect. That is, you have 4 years to succeed. If you fail, you're in deep trouble. Not only with the law, but also with getting money again. So that's what this says. And, but 4 years later, we had success. Okay? So I sent it back to the exact same study section, and they read the grant, and their reply was, "We're glad you didn't follow our advice." And that we were able to continue. But then I became a little bit even more audacious. I said, now I've done it in cells, now we're going to do mice. And that was also provocative because ES cells didn't exist. ES cells are embryonic stem cells, and they were going to be the source of making mice. We can use ES cells, put them back into, these are cells essentially that have no instructions of what to become. They're what we call pluripotent. They can become muscle, they can become skin, they can become teeth, hair, you name it. They have to be in the right environment and getting the right cues, and then they can become anything. Okay? They're undetermined. But put them back into the embryo, wherever they land, now they're going to get instructions and they participate in making the mouse itself. And now we're good enough that in the test tube, we can start with cells and directly make mice out of them. Okay? 100% of the mice will have that change. It was created in the test tube. Okay. So this is it. You say mice didn't exist, mouse ESLs didn't exist, but I did hear, first of all, what did exist were what I would call EC cells, embryonal carcinoma cells. These are sort of, they're really bad tumors. They grow, and they're capable of making all the tissues of an embryo, but it's completely disorganized. It's sort of gross that you'll see little patches of beating heart cells beating as if they were cardiac cells, heart cells. There are other places where you see hairs coming out, and they're making hair follicles. There are other places that you'll see bits of cartilage, and bits of bone, you name it. It's just jumbled up into an indescribable mess. But within that cluster were still these cells that were pluripotent, still capable of receiving signals and becoming anything. They're not committed. However, what was the problem with those is that they weren't capable of making the secret of life, and that is germ cells. Okay? Sex, that is sperm and eggs. They weren't capable of doing that, and that was critical because if we made just a mouse and it didn't have sperm or eggs, we couldn't grow up those mice. We couldn't breed them together to continue to make more mice with that same property. Okay, so every mouse would have to be created de novo, and then you could never repeat that experiment again except by starting all over again. However, I began hearing rumors that there was somebody in England that was trying to make EC cell-like cells. He at that point called them EK cells, but they were being derived not from a tumor, but from the embryo itself. Okay. And his name was Martin Evans. And so I heard about this rumor, so I called him up and I said, are you working on making EC-like cells, but then are coming from the embryo and thereby may contribute to the germline? And he said, yes. And I said, wow, can I come to your lab and learn how to work with these? And he said, sure. And so here we have two parts of the puzzles. I came in with gene targeting. He came in, he has mice, ES cells, now we call them. And together we could do something spectacular. That is, make mutant mice with any type of mutation possible. And I bring this up, this is Martin Evans, Sir Martin Evans now. In England, if you win the Nobel Prize, you become Sir. And what I'm trying to get across to you is that synergy is always better than competition. We could have each sequestered our own technology separate and gotten nowhere. We come together and then we can create gene targeting in mice. Okay? So the one take-home lesson is always two brains are always better than one, no matter whose brains you're talking about. So that's the applaud for collaboration. Now, let me give you how gene targeting is done. And you start with these cells. These are the ES cells that Martin Evans developed in his lab. And now they are capable of contributing to the germline. It's a colony of ES cells growing on feeder layers. And the feeder layers supply factors that keep these cells happy, but they're in limbo. Nobody's telling them what to do, and you can grow them, you can put them in the freezer, you can take them out again, and grow them up again. And it's in these cells that then we make the change. And then we take those cells and then stick them back into an embryo, that's shown here. We literally stick them into the embryo. This is a pre-implantation embryo. The cells at the bottom are the cells that are going to make the mouse proper. The cells on the outside will make the placenta. Okay? So we introduce those cells in a needle. We've changed the specific gene, and that's the only change present that differentiates the sources of those cells. Remember, 2 mice made the embryo, 2 mice made the ES cells. Okay? So when they come together, now we're making an embryo that it has 4 parents. Okay, clear? And what you can see is the cells going in, then they go into the inner mast cells, and then participating, making mice. And here is a stroke of bit of luck in this. The ES cells are embryologically a little ahead of the cells that are present in the embryo. Okay? Just a little fraction ahead. And therefore, the mouse that's coming out can be even 90% generated from the cells in culture, because they have an advantage. Often only 3 or 4 cells in that mass give rise to the embryo, but they're always going to be the ones, the ES cells. In fact, now we have technology that allows us 100% of the mice we generate coming from the cells in culture where we've changed a specific gene. And remember, with mice, they can do something that's really remarkable. You can breed brothers and sisters together for many, many generations, and they still survive. If we do that experiment, it fails, the embryo dies. In all other species except for rat, have they been successful in generating these cells, and even those cells aren't as good as mouse ES cells. Something that we still don't understand why mice can do this. I mean, it's quite remarkable that you can breed so many generations together. I should say the mice aren't perfect. Every strain of mice generated this way is different and carries about 1,000 mutations, which now are homozygous, therefore 2 defective copies. And therefore it's amazing that these mice can run around and breathe air and so on and so forth. But they do it, and we're lucky for it. So this is the actual process. So we have a pre-implantation embryo. We're injecting ES cells, which we modify the gene. Then we make a little slit in the back of the mouse that gives us access to the fallopian tube. We can introduce the cells there. Then they march into the uterus, continue to develop, implant, and then out comes a baby mouse with a changed gene. Okay? Whatever we've changed. That's the only difference, because these are inbred mice, so they genetically, they're all identical, but except for that changed gene. Okay, let me give you a couple of examples. We could take a gene from a bacteria, which in the presence of the substrate turns the cells blue. Or we could take it from a jellyfish, which makes a protein that fluoresces green. So wherever that gene is present, then all of those cells that are making are not going to be green. Okay? So if this gene is functional, in this case, in Vibrissa, the whiskers, then those whiskers will either be blue or fluorescently green. And that's what's shown on your slide. Okay? So you can see that this gene is important for making whiskers in a mouse, okay? Using either of those 2 technologies. That is the 2 markers. Okay. The same gene is also involved in making nails. So you can see blue nails, and you can also see green fluorescent nails. Okay? So they label that gene. So that tells us where that gene is working. Now the question is, what is it doing? It's obviously going to be in those structures. But how can we do that? Remember, you get 2 copies of every gene, one from your father and one from your mother. Now, if you have one defective copy, usually that's okay. So plus plus means two good copies, plus minus means one defective copy, and then minus minus, we just breed brothers and sisters together because we can do that with mice, okay? And now we have one-fourth of those progeny will have two defective copies, and what you can see, this mouse has alopecia. It doesn't make mouse hair, okay? Bald as bald can be, okay? So it also has defective nails, right? Come back. Ooh. Come back. Yep. It also has defective nails. Okay? So now we can go in the human population and ask, are there people that are bald and have defective nails? And they do. And what do we know about that? We know, in this case, we're going into a gene called HOXC13. And what we can see is in that gene, there is a mutation in the human. So that says whatever we found out in a mouse also directly is applicable to humans. And this has been done now thousands of times for every gene in the mouse, and that rule holds true. And even if it doesn't hold true, then it tells us something important that differentiates mouse from humans. So that's the first part of the story. Why we do it and what we're learning from it, and it's applicable, for example, to cancer. We can model cancer, childhood cancers, and then start understanding essentially what the mechanism is. And once you have an understanding of mechanism, then you have a chance to actually work out the cure. And you can use the mouse for that, to bridge that. Okay? You've generated a very specific defect in the mouse, you can cure it in the mouse, and then you can see whether that cure is applicable in humans. So now we go to early influences, the second embarrassing part of the story. And you always start with your mother. And this is a picture of my mother, age 19. She was a poet, but she's born a poet. She was writing poetry, she was publishing poetry. She went to the Sorbonne and was a lecturer there in literature and poetry. Then she got involved in politics, okay? And I'll come back to that story. But let me give you a little bit of her background. And it pertains to Portland. Okay? Her mother grew up in Portland. Okay? Her father was, actually, I even know the street, the shop, set up a hardware store in Portland and became a citizen, a known citizen in Portland. She talked her mother to leave her husband, and then, because she wanted to become an artist, so she talked to her mother to go with her, and they sailed around. This time there wasn't the Suez Canal. You had to go all the way around South America, ended up in Florence. And there she learned how to paint. And this is a painting of her. And this is a painting of my two uncles. In the middle is my mom. And the two uncles became physicists. And that's the littlest one became a really famous physicist. He developed things like the electron microscope, something that he was very proud of, television, no television in our house. But he had a mind. He actually went to Reed. All of you know where Reed is. He graduated there at age 14, went on to graduate school in physics, Cornell, and so on. So this was an ideal life. I mean, they had a villa, they had tutors to teach them the Greek, all the languages, art, you name it. And she had married a German archaeologist who was very wealthy, and they set up shop in Florence. This is another painting. This is a tea party, my uncle, and I mention him because I'll come back to that later. He actually brought me up once I came to the United States, and that's my mom. And I told you that she got politically involved, and she started confronting against both Nazism and fascism. And she knew that was short-lived. This is in the late 30s. So then, knowing that she was likely not to be allowed to continue her work, she moved up into the Tyrol in northern Italy, above Bolzano. And this is a chalet that she was renting. This is my mother in the foreground. And if you actually look very carefully, she was actually pregnant. So I was inside there. And what's remarkable is people recognized, once my name became known, they recognized where I grew up, and they sent pictures of the place I grew up, and it still existed. So they took us there, okay? And it's a house that looks very similar to a house that we own up in Salt Lake City, okay? That house overlooks 3 tiers of mountains, and this one looks over 3 tiers of mountains of the Dolomites. And my wife saw that. She says, "Oh my God, imprinting, who's swallowing Lorenz?" And here, I was buying that house in Salt Lake City just because I liked it. Little did I know that it was imprinted. But that life stopped essentially at when I was born, and then 3.5 years later. She would have continued her political work. This time she thought that she was in a place they wouldn't bother her, but they did. Gestapo came and took her off to Dachau, and this is a picture of Dachau. She was there for 5 years and survived, amazingly, because she's a beautiful woman, so you can imagine what she had to go through during that experience. This is when my mother anticipated that this was going to happen, so she had already worked out with friends that if the Gestapo ever came, then I would be sent to this farm, and that farm still exists. I went there. The same family still owns it. I was able to actually see everything. I could go to the chicken house. I knew where the chicken house was, and so on and so forth, which turned out to be very important knowledge later in my life, immediately after this. It turns out that after a year, all the money that she had given to them to take care of me ran out. And so they had to take care of their own family, so they sent me out into the streets. So at age 4.5, I was now going in the streets, started in Bolzano, and then started working down, so kept going south, because it was much better than going north, where there was a lot of snow and mountains. So I've gone there to the farm, to the same place where I grew up, and also to the farm where my mother and I grew up. And they still had saved all the poetry they had written when she was taken off to the Dachau. So they gave me that, all this collection. They kept it for almost 40 years before I saw it. Okay. So from after the war, after in 45, Dachau was liberated, and she is still alive. And then she searched essentially for me. It took her about 2 years to figure out where I was, because I had to keep moving. As soon as people know who you are and what you're doing, then you have to skip town, okay? Because I'm stealing all of my, I have to steal to get my food, and therefore your cover is blown. And so then you continue to move from one village to the next, and then finally ended up in Reggio Emilia, where she found me in a hospital, obviously with typhoid and malnutrition. But it took her about 2.5 years to make that connection. Then her uncle, her brother, my uncle, had started a commune in Pennsylvania. That says yes, good. And so you can imagine what, you're going from the streets, completely asocial situation where you took care of everything. Nobody's going to give you anything. Then to go to a commune, which was 65 families owning everything in common. So I go, no parents, to 65 parents. And they know everything. Okay? And they were terrified, and I was terrified, because they were terrified, for their daughters, and whatever their imagination could be with respect to my upbringing and what I could possibly be doing that could be anything good. So this is me about a couple of months after arriving. I still have the same outfit. I should say, when she found me, I was in a bed, and the bed was, there were beds lined up one after another. You know, just all the kids had typhoid, because if you're drinking any water, it was contaminated. Okay? And typhoid is sort of a strange disease. In the morning, you're fairly clear, and you can comprehend what's going on. Then by the time it gets noon, all of a sudden the fever kicks in, and you skyrockets, and then you're completely out of it. And then the cycle repeats every day after every day. And they didn't want me, when I was clear, they didn't want me to escape, so they took all my clothes away. So I lay naked in this bed, and all the other kids was there. And that's how you pass the time. And you know, I was always planning to leave, but by the time I was able to leave, I would be delirious. And my mom, what she did was to simply call different hospitals all over the country to say, does anybody by my name reside there? And she finally made a connection in Reggio Emilia, which is, you know, you could just go, it's essentially directly south of Verona, so you go through Verona and keep going down south in Italy. She made the connection. She arrived one day, and she knew my name. I couldn't recognize her. She had changed enormously, and also my recollection was when I was 3 and a half. And so, but she told me she was my mother. I said, terrific, free tickets to somewhere, it's better than here. And she bought me some clothes, and it's the outfit I'm wearing there. It's a Tyrolean outfit. You have a little cap, feathers in it, and the whole works. This is my uncle. He's doing what he didn't like to do, work on television, and was one of the developers of television at RCA. This is a school I went to, which is a Quaker school. And you know, in Quakers, what's important is service. You're born to be service. Okay? And so you want to solve world problems. I knew that world problems were political, so my first year of college, I was a political science major. And then I couldn't see where science integrated with political science. So I couldn't see, so I switched to physics. And this is the college I went to, and that was also a remarkable choice because it's a small liberal arts college in Ohio. And the feature is that you work a quarter, and then you study a quarter. You work a quarter, and you study. So it's a 5-year program all year round, but the jobs are related to whatever your interests are. So if you're interested in theater, you have theater jobs. If you're interested in law, you have a clerking job. I was interested in science, and therefore I had lab jobs. And lab jobs all over the country, not just at Antioch, but all over the country. And so then all of a sudden I end up at MIT when molecular biology was just being born. I mean, fantastic. And it was headed because, you know, you have no idea, you know, you have the influx of people from physics, mathematics, biology, medicine, chemistry, and so on, all coming together to start to look at biology in a very different light in terms of molecular terms. Okay? And it was just coming up. And you know, we thought we could solve any problem in the world. We had no idea how complicated biology is. And none of us had any training in biology. Okay? Nonetheless, we had rigor and imagination. And it was amazing, and no tools. I mean, when you consider what we have today, the tools were absolutely pitiful. And so, and this is still Antioch, and then from Antioch, I went to Harvard. And my mentor turns out to be Jim Watson. And all of you know who Jim Watson was because he's made the structure of DNA. That's what you were all involved in just a little while ago. And he's a mixed person in a sense. I mean, he has some terrible qualities, but he's really supportive. He's very imaginative and can really think about how biology works. I mean, he taught me not how to do science, but how to go into a field and then look at it and say, what are the questions that are interesting in that field that could be addressed with technology? And if the technology didn't exist, invent it. But then when I was there for a while, I was in the faculty at Harvard, and there were certain things that were missing, that were bothering me. One is that every day it was asked, what's new? And if you asked every day what's new, pretty soon you work on things that in a few days you have results. But I knew in the back of my mind what I wanted to do. I actually thought about gene targeting, and I knew I wanted to approach it, but I knew I couldn't do it in a few days. It took 10 years. And that kind of dedication probably wouldn't have worked in Harvard. The other thing that was important was synergy. Again, come back into that concept. At Harvard, they knew about competition, but they didn't know about synergy. Okay? And here, if I moved to Utah, now I had the luxury of one, being able to work at rival pace, and two, bringing people to there. It's a department that's just being born. So we could bring in people that would love synergy, love talking about science, having fun with science, because that's all it's got. We're paid essentially to solve puzzles. It's an amazing concept. Okay? People pay you to play. And this is my wife, and this is her first child. And this is simply a little later. And this is where we live. That's that villa that overlooks the 3 tiers of mountains instead of the Dolomites, the Wasatch Front. And here's what you can see from my window. In the spring, in the fall, and in the winter. You can see miles, literally 60 to 100 miles, just by looking out your window. And that has an effect on your psychology. It opens up that brain to be able to look at a lot of things as opposed to just what's right in front of you. And I think also had an influence. So finally, to the trip to Stockholm. And this is quite a trip. It lasts 12 days. And they start at 6:30 in the morning, and you quit at 3:30 in the morning. Every day for 12 days. It's exhausting. But it's beautiful. I don't know why, Sweden in the winter, and it's almost a polar cap. It's really cold. Really, there's snow everywhere, ice everywhere, but it's still beautiful. Simply because the ocean inundates the whole city. So wherever you're looking at, you always see boats and water. This is the hotel we stayed at. It's a magnificent hotel. I'll give you a couple stories from that. When you first arrive, first of all, they meet you at the airport in a limo. Okay? And that limo is yours for those over a week, okay, 9 days. And it's your driver, and he takes you everywhere. And you arrive at the hotel, and there's hundreds of people waiting for autographs, and everybody's in tune that the Nobel Prize winning people are coming there. 3 days later, Bruce Springsteen shows up at the same hotel. The crowd. Now you know what a rock star is. And it's not a scientist. This is a trio that won the Nobel Prize in 2007. Martin Evans in the center, who provided ES cells, and Oliver Spittis, who was thinking about the same things we were doing, but for entirely different reasons. He wanted to, still hasn't been done, he wanted to cure thalassemia. Okay? And he thought if he could change the mutation that caused thalassemia, then he would cure the disease. Obvious that it's taken a long time how to do it, and it's now just starting to begin to be worked. I should point out another thing about gene targeting, which is it's a thing that we should think about what may be coming, and that's CRISPR-Cas9. People knew about using the same technology we developed to try to cure diseases in humans, okay, gene therapy, using same DNA technology. But at that time, they were sort of immature as to how actually to do the experiment. And they did the experiment. They cured, this was bubble babies, they cured half a dozen kids. But they all died of leukemia. And it was predictable. Okay? They cured the disease, but the cells that they were growing up were simply the recipe for choosing cancer cells. Okay? And therefore, all of them got the exact same leukemia, and they all died. Okay? And that set the field back 20 years. And now it's starting to slowly come back, and now it is now being done rationally and with forethought, and therefore is starting to work. Okay? So it shows, if you jump into things sometimes without forethought, you may be doing a lot more damage than good. But I think we should think about that also with CRISPR-Cas9. People will offer, enormous possibilities, but we have to learn how to use it appropriately so we also don't do bad things. And that's where wisdom comes in. And I should say that the source of wisdom doesn't come from any group. No person at the top, no president, or whatever you can think about. It has to involve people from multiple disciplines, multiple backgrounds, all coming together and discussing the thing rationally. Okay, so here is fairly organized. The very first layer is essentially the recipients. There are 10 people roughly every year. Then back at them is 50 people that are involved in making that decision. Okay? And it's a very strange operation. They take all those 50 people, stuff them in a room, lock the door, and say, reach a consensus. Not popularity, whoa, consensus. And before they can get out, they have to reach a consensus. Therefore, it's unpredictable. The dynamics of 50 people trying to come to a consensus is unpredictable. And that's a good thing. But eventually, you come to a decision, and this year or that year, it's just this or that. This is the recipients right in the foreground, and behind them are the 50 people that are involved in. And the procedure is very, it's complicated in the sense that there's a lot of decorum. When you meet the king or the queen, you have certain things you're supposed to do. But in this case, it's pretty easy. All you do is watch the king. Okay, whatever he does, you do. If he sits down, you sit down. If he gets up, you get up. So that sets the stage. And then the king is there, the queen, she's a beautiful woman, also very a mixed background and very knowledgeable. And then the future queen is your right. And here is receiving the prize. And then afterwards you have a dinner. This is a small dinner of about 5,000 people. And it's orchestrated. It's amazing. There are plays going on. There's a big staircase that everybody comes down. And then all the servers come down. And the dishes have flares coming out of it. And they're initially all white, and then they turn pink, and then they turn red, and they turn, all the different colors. And by the time they reach the floor to serve you, they're all synchronized so that they're all the red flares. So it's orchestrated. They practice and practice until everything happens. And they do this every year, and they're very good at it because they've been doing it for over 100 years. And the reason it keeps on going forever is every night is a dance. And every night is a dance at a different college. And you have to dance with everybody. So you're dancing with 50 different people every night till 3 o'clock in the morning. It's quite a party. So I'll stop here and I'll open it up for questions. And I welcome any questions. The other thing that's really nice about this audience is there's a lot of young people. You are the future generation, and you're going to take the helm. I think we've been in charge. We've done a pretty poor job in certain areas, and now we're looking forward to you solving the problems we've created. For example, global warming. So I'd be happy to address any questions about anything, and the wilder the better. Yes?
Are genes owned by different
has been, that was a big problem, okay? As soon as somebody had a gene and they thought they knew what they were doing, they patented it, and then they did have a right to that gene. And that was a mistake, and I think people recognized that it was a mistake. You know, and the mistake was simply, it's everybody's gene, it's nobody's special gene. And so now you can't patent a gene. What you have to do is patent a process dependent on that gene. That's still patentable. And so, my feeling is that information, how science works, is really dependent on everybody having accessibility to it. Okay, nobody owns science. Good science is being done without any borders. It's done all over the world. Okay? So, and that's why it works. It's because of the input. You have no idea where the next bright idea could coming from. It could be Iowa, it could be New Delhi, you name it. Okay? We have no idea where the imagination comes up. But if it's made available, then everybody can utilize it. And I think so one has to be very careful with patenting. I think there are things that you should have priority if you put a lot of money into, and you have to if you're going to develop drugs for this or that. If people put in a huge investment, they need a return. But one has to work it out in such a way that they're not allowing other people to do that same thing, not the same thing, but a different thing, for example, with the same gene. So I think that making that distinction, not the gene, but how it's utilized, then will make allow that be not a problem. It's not perfect, but it's the best we can do. Yes? How much what? So I'll try to repeat the question. How much failure do you have? And I think, failure is a strange term. When you're an experimental scientist, what you want to do is learn from every experiment. Okay? If I do an experiment and it fails, and then I repeat it, it should get failed. If I repeat it again, it should get failed, and nothing's learned. But if you create the experiment in such a way that you have enough controls to tell you why it failed, then you can progress. So actually, you often learn more from the failure than the success. Okay? So in the science, it doesn't matter whether you fail or not. Obviously, we love the successes, but you're learning in the process, and that's what's important. So it's the process that allows you to go through, whether it fails or not, you're still getting information, and science is simply dependent on information. We're just creating new information. Yes?
Could you comment on the 2 gene-edited babies that were born and the one that's
Sure.
mom is still pregnant?
Sure.
What do you think?
And this is, you're referring to the twins that were born with a correction in the gene that's required for AIDS. They mutated mutated the gene that's required for the virus, AIDS virus, to be able to get inside the cell and do its havoc. And then you're resistant. And in fact, in this audience, there's probably several people here that are resistant to AIDS because they have that mutation. But the problem is we don't know what that gene is doing. Okay? But what you know is you're taking a good gene and making it defective and therefore resistant to AIDS. So you can argue, well, why not make everybody resistant to AIDS? And the reasoning is that gene is doing something important, or else you wouldn't be you wouldn't have it. Okay? So it's a cost, and we don't know right now. We don't know. We're beginning to get an idea of the cost, and it's actually going to have to be. It involves the brain, okay? And the brain is a very complicated thing, and so differences can make enormous differences in how functional that brain is. So the cost may be, may not be obvious, but it's there. And the other possibility is that that same gene is involved in accepting other different viruses. And so you may be changing your susceptibility. You may now no longer be susceptible to virus A, but now maybe become susceptible to virus B. Okay. So until you know the full extent of what that gene is doing. And I would say in humans, that's a very difficult thing to evaluate. I think you're playing with fire. And so, obviously the children didn't die, okay, so that's a good thing. That would have been the worst thing, okay? And they were pretty sure that wouldn't happen. But there's another problem also. In the process of changing that particular gene, you may have side effects that you're changing genes that you have no idea what they are. Okay? It's not specific just to that gene. And we know that actually often happens, particularly with CRISPR-Cas9, that system that you utilize. So it's likely that those children will have other mutations which may be deleterious and which won't show up till later in life. So I think it's, and I think the Chinese have taken the appropriate approach, and that is, this is not acceptable. We're not ready for this type of technology to be used in humans, and they've really pushed hard on that. So I think, it's again that same situation with gene therapy many years ago, that they had good intentions, but they also had a bad outcome. And I think the same thing may happen in these cases until we really understand. And the one thing I should say, you can do gene targeting in the body, or you can do it in cells, and then in cells put those defective cells down back in the body. Okay. That's the main. You should only do that experiment, because in culture you can exactly look to see what you've done. Have you done what you expected? Have you not done something you didn't expect? You can sequence the whole genome if you have to, and see exactly what's happened, and then put it back into the body once you know exactly what's happened and know what the risk factors are. But even under those conditions, I would say, I have no problem in making any change I want in a mouse. My daughter does, but I justify this because I try to do something and there are things I won't do. And that's all simply decision making. People have to reach their own conclusion. What are they willing to do? What they aren't willing to do. There are many experiments that I wouldn't do. I wouldn't do things in primates, non-human primates, simply because I look at them and they look a lot like me. Okay? And I don't know how close that resemblance is, but it's likely to be much closer than we anticipate. I think our mice can recognize faces, human faces. Okay? So their intelligence isn't that bad. So even working with mice has problems associated, but you have to counter it with what good you can do with it, and to try to do things that are A, good, and B, aren't devastating to the mouse, at least for over a long period. Way back there?
Yeah. So, in your work, how much do you partner with computer science people? I'm curious.
How much do we partner with what?
People who specialize in computer science.
With other scientists?
Computer science.
Somebody?
Computer science. Computer science.
Oh, how much do I think about future science? Future science?
How much do you partner with people in computer science?
Computer science.
Oh, computer scientists. Sorry. No, a lot. I should do more. Because, computers have changed science irreparably. We can handle information in ways that was not conceivable before that. And so, I just went to, in this city, in this city, I went to their genetics department, and I would say 80% of those people were trained in computer science. It's that, it has enormous influence, and it's incredible, what you can do now with a computer that you couldn't even imagine a few years ago. Just analyzing a genome. That was inconceivable even 10 years ago, or 15 years ago. Okay, so I think it's changed enormously, and it keeps changing. And you can see the progress of computer science, and you can parallel yourself going along that same trajectory because you're dependent on it. as soon as new things come along, you have to embrace it. I mean, one thing about science is it's uncomfortable. You know, human beings love to sit in front of a TV, potato chips in one hand, beer in the other, and simply taking it all in, and then tomorrow you've forgotten it all. Okay? That's heaven. In science, you have to be uncomfortable. You have to, be on the edge, because that stimulates what's going on up here and drives you forward. And so we have to accept change rather than try to reject change. And computer science is the one thing that we follow as quick. As soon as a new development comes along, bam, we grab it, and then waiting for the next bite and so on. Yes.
Do you think the time you spent on the streets of Italy influenced your path as a scientist?
Yeah. No, I've thought about that question, and I think so. I mean, I can see aspects of our lab that came from that. One is, I'm pushing collaboration, but our own lab, whatever it's doing, learns to do everything about it. I mean, for example, if we have to do histology, we could get better histology, send it off somewhere and get the results back, and they could do it cheaper. But I insist on them doing the histology themselves, because if they're looking at the slides and they're comparing normal and mutant, they're going to look at those slides much more aggressively and in finer detail than if they had sent it off somewhere else and they'd be told the results. Okay? So self-sufficiency, in the sense that I want the student to learn is with their own hands all the processes. And so I think that's important to me. And I could see, and the only way I existed was to be self-sufficient and be good at it. Okay? I had to become a good thief in order to survive. Okay? And I became a really good thief. I mean, I could, but the curious thing is, they say, well, pickpocket. That's useless. Money, if I had money, the first thing I would go, I'd go to a store and give them money, they'd call the cops. So that wasn't a choice. What I wanted is food, and I hadn't eaten a hot meal until 9 years old. Okay? It was all raw food. That's because that's what's available. But I knew where the chicken house was. I could find eggs. I knew lots of stuff, and that allowed me to survive. And again, I sort of stressed that most of those kids died. Okay, you're selecting out the ones that could survive.
Maybe 2 more questions.
Sure. Over here.
Did you ever, can you target genes of mice, not just the embryos, like change of mice that are alive? Can you change genes in adult mice, or can you only do it in embryos?
Right. Good question. And what we can do is we can do it both ways, in the sense that we can not only create a mutation in the germline so that every cell in that body has that change, but we can make it in such a way that the mutation can then be restricted to a particular cell type. Okay, so we can choose that cell type, and then we can also restrict it to time. So that could be in the adult. For example, we can put the mouse through a learning paradigm, and then it activate the gene before, middle, and afterwards to see the transition essentially from setting up a memory to then accessing the memory. So you have fine control as to when, where, and how you mutate it. Further, you know exactly what you want to change. So if you see a mutation in humans that gives rise to synovial sarcoma, we can make the exact same mutation in the mouse, and we can restrict it to the same cells that give rise in humans, and we can even restrict it before, after birth, and so on. So you have all of those capabilities. Yes?
So now, are you still active in research? Are you still
Yes.
working on it?
I'm very active. In fact, I'm looking for a second life. Though I wasn't satisfied with one lab, so I'm going to open up another one. And both of them are going to be 100%, but that only doubles the load. No, I think, my My wife tells me I'm going to die in the lab, and that's not a bad thing.
Well, thank you so much, Dr.
I just want to take a moment on behalf of St. Mary's Academy to thank our sincerest gratitude for having Dr. Capecchi here today. But I also want to thank our host for this event, Dr. Charles Keller, and the Children's Cancer Therapy Development Institute, and all of their kindness and generosity in bringing Dr. Capecchi here today. So thank you all again. We look forward to seeing you at our next lecture.
Also thanks to Charles, but also to the audience. I think it's, I know it's Saturday, there's lots of other things you could be doing.